Hi everyone, it's Dr. LeBlanc with Unit 9 on Genomics, Biotech, and Medicine. This is Lecture 9A, Genome Organization. Our learning goals are to learn about the different types of genetic maps and their usefulness, to understand what cytogenetic, linkage, and physical maps are, to understand fish, to understand how linkage maps rely on genetic markers, to understand how STSs surrounding microsatellites are used as genetic markers, to understand how RFLPs can be used as genetic markers, to understand how next-gen DNA sequencing can be used to create physical maps and how it can be used in many other applications, to understand how to go from sequence to annotation by analyzing a physical maps, and to understand the future of genomics in medicine and agriculture. This is a picture of chromosomes at metaphase on a microscope slide. They have been hybridized to a probe, a DNA probe that's been fluorescently labeled. The DNA probe contains the sequence of telomeres for this particular species, and you can see that every single chromosome on the slide has a fluorescent dot at the end of the chromatids. That is because the sequence of telomeres for any given species is the same at the ends of all the chromosomes. Genomics, or the molecular analysis of the entire genome of a species, has three main phases: mapping, sequencing, and analysis. There are three types of maps that can be made from a genome: a cytogenetic map, a linkage map, and a physical map. The results of each of these types of mapping techniques might be slightly different because the resolution is different. Cytogenetic mapping consists of staining chromosomes and looking at them through the microscope. There are different types of stains for chromosomes. GEMSA is one of them. Each of the different stains will yield a different banding pattern for a particular chromosome, but that pattern should be the same for no matter what the source of the DNA is, if you use the same stain. Genes are mapped using cytogenetic mapping relative to the bands on a given chromosome. Using lots of different chromosomes from different individuals in a population, you can map phenotypes to particular banding patterns on a chromosome. The CFTR gene is the gene that, when mutated, causes the condition in humans known as cystic fibrosis. It is a transmembrane regulator. It regulates the passage of chloride ions through the membrane. The gene is located on the Q arm, which is the long arm of chromosome number 7 in region number 3, towards the end. All chromosomes are given a designation of P for the short arm and Q for the long arm, with the centromere in the middle. Here's another fish picture, fluorescence in situ hybridization. Notice that in this picture, only 2 of the chromosomes are showing fluorescence for 6 different probes. These probes all came from different regions of chromosome 12, and so the 2 chromosomes you are looking at here are the 2 chromosomes in the homologous pair of chromosome number 12. These probes are exclusive to chromosome 12 because they don't hybridize to any other region in the rest of the genome. Linkage mapping relies on genetic crosses. The genes are mapped relative to each other, and the distances are computed in something called map units or centimorgans, which relate to recombination frequencies. The further apart the genes are on a chromosome, the higher the recombination frequency is going to be between them, and the further away in map units those 2 genes are going to be placed on the chromosome. So this is a segment of Drosophila, chromosome. This is the skewed gene, and this is the white gene. They are 1.5 map units apart. Linkage mapping can also be undertaken with short tandem, sequence tag sites containing microsatellites or STRs and RFLPs. RFLPs are restriction fragment length polymorphisms. Here we have a segment of a chromosome that is showing all the different sites where EcoR1 restriction enzyme will cut the DNA. We have DNA from Thelma and we have some DNA from Louise. And for most of this segment from both of these individuals, the EcoR1 sites are in exactly the same position. However, Thelma exhibits an additional EcoR1 site in between these two sites right here. So Thelma is actually homozygous for this EcoR1 site that cuts the DNA here. Okay. Louise is also homozygous because she's missing this EcoR1 site right here and here. So when Thelma's DNA is cut with EcoR1, we get a fragment in this particular region of the chromosome that is 1,500 base pairs and another one that's 3,000 base pairs. Okay, and that's the same for both copies of this chromosome, a 1,500 and a 3,000, because both of these chromosomes have this EcoR1 site right here. Louise is missing this EcoR1 site, so the fragment that's generated in this region of the chromosome is 4,500, which is the same as 1,500 plus 3,000. Okay, and she has the same pattern on both of her chromosomes in this region. This is missing the EcoR1 site and missing the EcoR1 site here. So both Thelma and Louise are homozygous for their particular patterns. Bob, on the other hand, is not. Bob is heterozygous because one of the copies of the chromosomes that he has this EcoR1 site here. And when you cut this region of the chromosome, you'll generate a 1,500 base pair fragment and a 3,000 base pair fragment. But the other copy of the chromosome is missing that EcoR1 site. And so the fragment that he will generate from this region of the chromosome is 4,500. So Bob's DNA, when you cut with EcoR1, is going to have this 1,500 base pair fragment, a 3,000 base pair fragment, and a 4,500 base pair fragment. So if you look at the DNA from the three of these different individuals, you will find that Thelma has this site, this fragment, and this fragment. Right? She has the 3,000 base pair fragment and the 1,500 base pair fragment. Louise, on the other hand, has this 4,500 base pair fragment. Okay, but she does not have the 3,000 and the 1,500 because there's no EcoR1 site in the middle of this segment of DNA to cut this into two pieces. Remember, both Thelma and Louise were homozygous for their particular patterns. Bob, on the other hand, has one chromosome with that extra EcoR1 site in it and one without. Okay, so he's got the same pattern as Louise from this chromosome and the same pattern as Thelma from his other chromosome. Okay. Most of the fragments you notice are the same. Many of the fragments are the same in all three of these individuals. When these fragments are the same, they're called monomorphic. Okay, but we're mostly interested in the polymorphic sites. Genetic mapping can also use sequence tag sites that contain polymorphic microsatellites or short tandem repeats. This is a diagram showing the same locus on chromosome 1. On the chromosome that you inherited from mom, you have several tandem repeats At the same locus on the chromosome that you get from dad, you only have a few. So if you use the same primers that will bind to the flanking regions around this microsatellite, okay, this will generate a long PCR product, and this will end up yield a short PCR product. So when you analyze the DNA from the PCR reaction on a gel, you should end up with two different lengths of PCR products. A long one that reflects the fact that this site on the chromosome that you got from mom has many copies of this repeat, and the shorter one that reflects the fact that this microsatellite on dad has fewer repeats. So let's look at this again. When you have a set of chromosomes and you add PCR primers that are specific for just one locus on, for example, chromosome 2, those primers are going to stick to the DNA, the same locus on chromosome number 2, and flank this microsatellite. When you perform the polymerase chain reaction and run the products out on a gel, the long microsatellite will give you a big PCR product, and the shorter microsatellite on the other chromosome will give you a shorter PCR product. It has fewer repeats. So how can we use this in paternity testing or forensics? Well, here's a pedigree. Girls are circles, boys are squares. So this is mom, and this is dad. And this is offspring 1, 2, and 3, or daughter number 1, daughter number 2, and son. If we use a polymerase chain reaction on mom and dad, we see that mom has two different alleles, allele 1 and allele 2. So she's heterozygous at this particular polymorphic locus. Dad is also heterozygous, but has two different PCR products. It's shorter, 3 and 4. Okay, so all of the offspring, if they indeed are the offspring of these two parents, should have one allele that they got from mom and one allele that they got from dad. Let's see if that works out. Individual number 3 is a daughter. She got this allele is the same as this allele from mom, and this allele is the same as this allele from dad. So she's got one allele from mom and one from dad, so that everything seems to be working out just fine. Let's look at this daughter. She got mom's other allele, a slightly shorter one, and the same allele from dad. So indeed, she also has an allele from mom and an allele from dad. And the son as well has the same allele from mom, but he's got the shortest allele that dad had. A while ago, I created 21 polymorphic markers from human chromosome 12. This is the list of all the different markers I created. These are the loci that match up to the different banding patterns on the cytogenetic map. And this is the primer sequence that flank a microsatellite. And the microsatellite that I identified the markers around are CA repeats. CA repeats are very prevalent in the human genome. Millions of them exist on all the different chromosomes. And if they're long CA repeats, they tend to be polymorphic. Okay, so these were the sizes of the PCR products that are generated using these primers. Okay. And when I looked at a very small population, these are the number of alleles I found, and this was the heterozygosity. The heterozygosity is the percentage of individuals in a population that are heterozygous for that particular PCR product. So if you look at one of these markers and you look at DNA from 10 different individuals and then some controls, you can determine the heterozygosity. So if we look at each of these individuals, if you see two bands, then that individual is heterozygous. It has one length of CA repeat on one chromosome and a different length of CA repeat at the same locus on the other chromosome. So here's a heterozygous person, heterozygous, heterozygous. All of these are heterozygous except for the last two. These are not. These are homozygous. So 8 out of 10 gives you an 80% heterozygosity, which is pretty good, especially in this small population. Let's count the number of alleles. All you have to do is look at the different sizes of PCR products that are generated. So here's 1, here's 2, here's 3, and here's 4. Yep. 1, 2, 3, 4 different alleles, 4 different lengths of PCR of microsatellites were detected by the polymerase chain reaction. It also is kind of interesting in this population that it looks like an allele that's supposed to be here is missing. So if we looked at a larger population or a different population, we might actually be able to find 5 alleles instead of just 4 or even more. Students at Merrimack College are undertaking a project to identify polymorphic markers for the Self-and-Moly. Self-and-Moly is an emerging model system for population studies and for environmental studies and for evolutionary studies. It's closely related to Linkage, and they are creating several different polymorphic markers to be used in those types of studies. What about physical mapping? Physical mapping relies on DNA, actually not cloning anymore, but sequencing techniques. The genes are mapped relative to each other based on base pairs, the actual number of base pairs. The old method to determine the sequence for a given genome was to take the DNA, process it, purify it, and clone it into vectors, and then sequence the DNA from the vectors and then assemble it into a contig. So you end up with lots of different clone sequences that are overlapping, and you can assemble them based on the sequence similarity by the ends. Next-gen sequencing methods rely on assembling contiguous sequences, but they skip the cloning step, which is the most tedious of all. So if we look at the workflow for next-gen sequencing, we start with some kind of tissue, either some blood or some saliva or some fossils or some other tissue. And the genomic DNA is either you have genomic DNA or you reverse transcribe some RNA to get some cDNA, or you have some chromatin or some other DNA preparation from a particular experiment. The DNA is fragmented either by shearing, physical shearing. You could shake it, pipette it up and down to shear it to uniform sizes. Or you can cut it with enzymes to create some overlapping sequences. The next step then is to add an adapter to the ends. And you add different adapters to the ends based on their 5' or 3' ends. And the next step is the most unique step for next-gen sequencing, and that is to immobilize the DNA onto some solid matrix support, either beads or slides, and then perform the sequencing reaction. So one of the most popular companies these days, one of the companies that's having the most success is Illumina, and their method is called sequencing by synthesis. So at the end of the last slide, we saw that the DNA was attached to a solid matrix. Okay, it's attached to a flow cell or a bead or some kind of solid matrix down here. And you have the adapters at the end, and what you do is you add a primer to it. Okay? The primer has polarity, okay? And you're going to sequence the DNA based on this template using a DNA polymerase in a 5' to 3' direction. And you add the nucleotides one at a time, and they get synthesized into a short fragment of DNA. Now, these nucleotides are modified to contain fluorescent dyes. Each of the nucleotides has a different fluorescent dye attached to it. An optical sensor both excites and senses the light emitted by this fluorescence, and the computer, it's detected, and the computer then records which nucleotide was just added and gives you a readout. Okay, then you can cleave the fluorescence and wash it away and reuse it again and again. Repeat. Okay, here's another diagram that shows something similar. So what you end up with is a sequence of nucleotides based on that process. Once you get all those nucleotide sequences, then you have to do some matching up. These are short reads, but when you have so many different fragments and they're all overlapping, you can then assemble one long sequence just like this, looking for regions that overlap. Okay? If you do many reads from the same section of the genome, they should all be the same, but sometimes they're not. Sometimes you have some ambiguity in it. And you have to take that into consideration, and you come up with a consensus sequence. What's the most popular nucleotide at this position? Well, you can easily see that it's G and not this T. So this was an aberration. And you could do this over and over and over again, and if you have a reference genome sequence, you can compare it to that. If not, you just get a consensus sequence from a newly synthesized or newly sequenced genome. So after you have the sequence, then you have to analyze it. And that is careful examination and annotation of the sequence of nucleotides and of genes and genomes. And annotation, what do we mean by annotation? Well, you look for functional sequences. You look for promoters. You look for start sites. You look for stop sites. You look to see if the sequences have any homology to any other sequences in the databases. The students at Merrimack College are involved in a project where we isolate new bacteriophages and have them sequenced, and then we annotate them. This is in collaboration with the Howard Hughes Medical Institute and the Science Education Alliance. So far we have 4 genomes that we've annotated. This is an excellent overview of what a segment of a chromosome looks like. If you were to fly over this spot on chromosome 11, you get to see genes, you get to see pseudogenes, you get to see microsatellites, you get to see repetitive elements, you get to see what a chromosome looks like. Also, another ancillary video to the one that we just to this one is a talk by Richard Resnick, who was a CEO of GenomeQuest. When he made this video, he predicted what the 2016 presidential elections might be looking at as far as genome sequences are concerned. And you can, if you watch the video, you can see whether or not his predictions are true. But this is an excellent, excellent video to put some perspective on sequencing genomes. So in summary, physical, linkage, and cytogenetic maps give different kinds of genetic information. By looking at banding patterns, breakpoints, and other variations in chromosome structure, specific genes can be linked to a region of a stained chromosome that can be observed through the light microscope. Fluorescence in situ hybridization can localize a particular DNA sequence to a specific spot on a chromosome visually, so you can see it through a microscope. Restriction fragment length polymorphisms can be used to match DNA samples. Sequence tag sites that contain polymorphic microsatellites or short tandem repeats can be used to locate genes for specific linked traits in forensics and in medical genetics. Heterozygous individuals have short tandem repeats with different lengths at the same sequence tag sites on each chromosome of a homologous pair. This is detectable by the polymerase chain reaction. An STS containing an STR, a sequence tag site containing a short tandem repeat, can have many different alleles that vary by a single repeat length. Good polymorphic markers have a high heterozygosity, which is the percentage of people in a population who are heterozygous for the locus, and a large number of alleles. The more variability, the easier it is to match a specific sample to a specific individual. Physical maps contain information about the nucleotide sequence of genomes. Next-gen sequencing methods fragment genomic DNA, add adapters, tether the DNA to a solid matrix, and determine the sequence by synthesizing DNA using the fragments as templates. The nucleotides that are added are labeled by fluorescence. This is referred to as sequencing by synthesis. Detailed analysis of DNA sequences is referred to as annotation, where function is assigned to a specific region of a chromosome down to the nucleotide level. Easily obtainable DNA sequence information has revolutionized the fields of medicine and biotechnology, as we will see.